First, we have the stages of neural impulses. The first stage is depolarization. In depolarization, a stimulus starts the depolarization of a membrane. Depolarization is also referred to as the upswing, and it is caused when positively charged sodium ions rush into a nerve cell. As these positive ions rush in, the membrane of the stimulated cell reverses its polarity so that the outside of the membrane is negative relative to the inside. Next, we have repolarization. Once the electric gradient has reached the threshold of excitement, the downswing of repolarization begins. The channels that let the positive sodium ion channels through close up, while channels that allow positive potassium ions open, resulting in the release of positively charged potassium ions from the neuron. This expulsion acts to restore the localized negative membrane potential of the cell, bringing it back to its normal voltage. This process of depolarization, repolarization, and recovery moves along a nerve fiber from neuron to neuron like a very fast wave. While an action potential is in progress, another cannot be generated under the same conditions.
Now moving on to the stages of the sliding filament theory. A nerve impulse arrives at the neuromuscular junction, which causes a release of a chemical called acetylcholine. The presence of acetylcholine causes a depolarization of the motor end plate, which travels throughout the muscle by the transverse tubules, causing calcium to be released from the sarcoplasmic reticulum. In the presence of high concentrations of calcium, the calcium binds troponin, changing its shape and moving tropomyosin from the active site of the actin. The myosin filaments can now attach to the actin, forming a crossbridge. The breakdown of ATP releases energy, which enables the myosin to pull actin filaments inwards, shortening the muscle. This occurs along the entire length of every myofibril in the muscle cell. This is known as a power stroke. The myosin attaches from the actin, and the crossbridge is broken when an ATP molecule binds to the myosin head. When the ATP is then broken down, the myosin head can again attach to an actin binding site, further along the actin filament, and repeat the power stroke. This process of muscular contraction can last for as long as there is an adequate amount of ATP and calcium. Once the impulse stops, the calcium is pumped back to the sarcoplasmic reticulum, and the actin returns to its resting position, causing the muscle to lengthen and relax.